Okay, so a couple announcements before we get started on this. One of the groups just brought to my attention that there are some funky things that got lost in the bowels of Brightspace. So I'll make sure that the two remaining problem sets you don't have graded, one from transport a long time ago and one on DNA. I'll make sure those are updated by this evening. I'm also having a couple people saying that something weird is going on with their quiz number 12. It's not everybody, so just take a look at if that's what you remember that your grade should be, and if it's not, let me know because I'm going to look into those individually. Finally, there is an item in the grade book about a discussion post on your exam reflection, but we made that an in-class assignment instead, so don't worry about not having anything in there, and I will figure out how to get that deleted from the grade book. Does anybody have any other questions about what's going on in there? You should now be seeing that your lowest problem set has been dropped. Crossing my fingers, but when I double checked, at least on my end, it looks like it has been.
My end has been too.
Okay, great. Okay, if anybody else finds anything funky in the grade book, let me know as soon as possible, because I know it can be a little anxiety provoking not to know how you're doing, and I want to make sure we're all squared away. Okay.
If we do have that problem on quiz 12, do you want us to chat you or just like send you an email, or?
Just send me an email so that depending how long it takes to figure out what's going on, I have a record, even if it's after class. Okay. So hopefully everybody got something. Okay. Hopefully everybody got my message yesterday afternoon about the videos, but if not, I want to make sure that we at least have a high level understanding of what's going on with fermentation and the TCA cycle before we break into that with the problem set. So when we ended on Friday, we were talking about glycolysis, which is the first step of cellular respiration. And after glycolysis, what happens is that we have like basically a checkpoint. So this is the point at which we're going to determine whether we continue with the rest of aerobic cellular respiration or whether we go into a different process called fermentation. So fermentation is not a step in aerobic cellular respiration. It's like a separate pathway, but it also starts with glycolysis. And so how we decide is whether oxygen is present. So if it is, we do pyruvate oxidation, and we're going to continue with the process that we'll be finishing up this week. If there is no oxygen, the cell is going to do something called fermentation. And the purpose of fermentation is really to regenerate that NAD+ that was one of our inputs for glycolysis. Because glycolysis needs glucose to happen, but it also needs NAD+. And if we use that all up and convert it all to NADH and never return it to its original form, we can't do glycolysis anymore. Okay. So if we are going down this different separate pathway because we don't have oxygen, we're trying to regenerate NAD+. And the way we're going to do that is by basically breaking down our pyruvate into something else. So depending on what type of organism or what type of cell we're talking about, we're either going to turn pyruvate into lactate. So this is called lactic acid fermentation. And the idea is that the presence of lactic acid is one of the things that makes like your muscles burn when you're exercising really hard. The purpose is to take that pyruvate we end up with at the end of glycolysis, we reduce it to lactate, which means that we oxidize NADH back to NAD+, right? Because NADH was our reduced form, NAD+ is our oxidized form. So we take that NADH that we also ended up with at the end of glycolysis, return it to this form of NAD+, and then we can reuse it so we can keep doing glycolysis. Ethanol fermentation works exactly the same way, except we produce ethanol instead. That's how we get, alcohol fermentation in beer or in bread, or a lot of different types of fermentation we see in kind of food products. Okay. So this is moving back to our normal pathway if there is oxygen. So fermentation is like a side step if we don't have oxygen. Now we're back in the main pathway. So if we're back in the main pathway, we shuttle pyruvate into the center, into the matrix of our mitochondria. Right, so it goes through the outer membrane, it goes through this intermembrane space, and it goes through the inner membrane. Okay. And after it's transported here, we're going to take off a molecule of CO2. Now pyruvate now is losing a CO2, so we're going down a molecule of carbon here, right? And we attack on this molecule called CoA, and so we end up with something called acetyl-CoA at the end of this process. As we're doing this, we also do form another molecule of NADH for each pyruvate molecule. So here we have the equation for what that would look like. Start with 2-pyruvate. In order to get NADH at the end, we had to put in some NAD+. So we get 2-acetyl-CoA, 2-CO2, and 2-NADH. Now this is all 2 because we broke the glucose into 2 pyruvate molecules, remember. So that's why we have basically double of everything here. Then we go into the citric acid cycle, which is also called the Krebs cycle, which is also called the TCA cycle. All of those things mean the same thing. This is happening in the mitochondrial matrix, which is good. That's where we already were with our pyruvate. All right, so we're starting with acetyl-CoA. We're entering the cycle by attaching that acetyl-CoA molecule to something called oxaloacetate, which is also called citric acid eventually. So in a series of steps, we're going to release a bunch of stuff. The main point of the citric acid cycle is to create electron carriers, these reduced electron carriers. So we do create a little bit of ATP, but it's mainly to produce these electron carriers, which we're going to use in our final step, the electron transport chain and oxidative phosphorylation. Okay. So the cycle looks like this. So here on the left, we see our acetyl-CoA coming in. It's a 2-carbon molecule. Pyruvate was a 3-carbon molecule, but we pulled off a carbon when we got rid of carbon dioxide. So it's joining with oxaloacetate. It's going around in this cycle. We're producing our very important reduced electron carriers, NADH. FADH2 is another one. We have fewer of them, but it works about the same way. We're getting rid of CO2, and we're creating a molecule of ATP. This gets rid of all the carbon from our original glucose molecule. So after this, there's no carbon left. And the other thing to remember about the citric acid cycle is because we have 2 acetyl-CoA molecules, this is just showing one cycle. So this would happen twice because we have 2 acetyl-CoA molecules because we broke glucose into 2 parts way back in glycolysis. Okay. And so here's the final equation. We can see that our main thing that we're producing here is those reduced electron carriers. Okay. So your problem set, which you will find here, asks you first, we're going to talk about this process of pyruvate oxidation. So this whole first page is about pyruvate oxidation, where these different steps are going. We're thinking critically about what all of these parts are doing, and counting some carbons and so on. And then the second page, we're thinking about the citric acid cycle. And I've pasted the picture in here so that you don't have to keep flicking back and forth. We're not trying to memorize all the steps here. What we're really interested in is mainly the inputs and then what's coming out. Okay. So we're going to go ahead and do that. So just note that fermentation isn't on this problem set. What we're tracking here are 2 steps that are part of aerobic respiration. Fermentation is a separate side step, so it would not be a step in this aerobic cellular respiration pathway.